O-Level Biology 5090, Chapter 5, Enzymes. And what are enzymes? Enzymes are protein in nature and act as biological catalyst. And what are the biological catalysts? And catalyst is a substance that speed up a chemical reaction but is not changed by the reaction. It means it is not changed during the reaction, so it can be used again and again. But keep in mind the different types of enzymes are required for different types of reactions. So enzymes catalyze all types of metabolic reactions, whether they are catabolic or whether they are anabolic reactions, because they work in a very special way. They lowers down the activation energy of the reactions. So what is the substrate? Substrate is that substance on which the enzymes act. On which the enzyme act. Enzymes catalyze chemical reactions involving the substrates. In case of single substrate, the substrate bonds with the enzyme active site and an enzyme substrate complex is formed. That is, you can see over here, enzyme, and this is substrate. And substrate will always join on the enzyme which has this depression-like active site. So enzyme will move into it and there will be the formation of enzyme substrate complex. Enzyme substrate complex. And substrate can be single or more which can participate. So different types of reactions can take place with the help of these enzymes. And now I would like to give example of substrates. They can be the nutrients, carbohydrates, lipids, or proteins. So substrates can be any one of them. I would like to move towards the next. And here, more information about enzymes. Speed up the chemical reaction required in very minute amounts, very small quantities required for the process. But they can be, you can say, obtained at the end of reaction to reuse in the other reactions. Are highly specific in their action. Highly specific means the different enzyme for different substrates. So you can say the enzyme which is for carbohydrates cannot be used for proteins. So the different enzymes for different nutrients are affected by temperature and pH. At high temperature, they are denatured. They become shapeless because at that time, the energy of the molecules is increased. They are activated. So substrates, you can say, when strike with the enzyme very fastly, the shape of the enzyme is changed. And that is, we say that the enzymes are denatured. Same happens at different pH. The enzymes are not working properly. They are denatured at very different pH. As for example, if they are like to work on acidic pH, they don't need neutral or alkaline pH. So they will work best in their uh, acidic conditions. So it was all about the enzymes. We will move next. Here is an example of an enzyme, which is catalase, which is nearly found in all living organisms, bacteria, plants, and animals. If exposed to oxygen, it catalyzes the decomposition of hydrogen peroxide to water and oxygen. So this is very important enzyme because in this way it protects the bodies from oxidative damage and reactive oxygen species. So in this way, catalase is very helpful for the living organisms to save them from damage. Now I would like to move towards the next slide, in which we will discuss one hypothesis by a scientist. And the hypothesis is called as lock and key, which is very important. Lock, what does it mean? Lock, which we use at home to lock our doors, and key is used to open the doors. Key always fits into the lock. Lock will not fit into the key. Key is used to slide into the lock to open it. So here in this case, the lock will act as an enzyme and key will act as substrate. Because always substrate will go to join on the enzyme, because enzyme has this active site, which is specific, and specific substrate will join with it. So there will be the formation of enzyme substrate complex. So this is the depression or the shape which is formed on the enzyme, that is called as active site. So only specific enzymes will open up. Okay, next, lock and key hypothesis in the form of this enzymes. You can see red color enzyme, and you can see a substrate over there. And this substrate will, when joined with the on the active site of the enzyme, there will be formation of enzyme substrate complex. And then after some time, you can see the enzyme remains the same and it is not used up in the reaction, and there is formation of two products. Sometimes there is formation of product or products. In anabolic reactions, product is formed, and in catabolic reactions, products are formed. Now I would like to move towards the next type of reactions which are catalyzed by these enzymes. First of all, type of anabolic reaction. As an example, here you can see this is an enzyme. You can see the shape of these two substrates. Two substrates, they will fit into it on the active site of the enzyme, and there will be the formation of this enzyme substrate complex. So it means they are specific in nature. Any other shape cannot be entered or attached on the active site of this enzyme. So only the specific shape can be attached, or specific substrate can be attached on the active site of the enzyme. So here you can see enzyme substrate complex, and after this you can see formation of this product, from two substrates. These two substrates are changed into one product. So this is an example of anabolic reactions, which is called catalyzed by an enzyme. Now I would like to move towards the next type of enzymatic reaction, which is an example of catabolic reaction. And you can see over here, one substrate and one enzyme, they will join together. The substrate is attached on the enzyme active site, and then there is formation of products. Here there will be formation of two products. Enzyme will remain the same, and this is in the previous example, there was the formation of one product from two substrates. Now, one substrate is changed into two products. So this is type of catabolic reaction, which is catalyzed by the enzymes. Now, I would like to show you effect of temperature on enzyme activity. How do the temperature affects the enzymes? Like most chemical reactions, the rate of an enzyme catalyzed reaction increases as the temperature is raised. This is obvious. When we increase the temperature, the rate of reaction is increased. A 10 degrees centigrade rise in temperature will increase the activity of most enzymes by 50 to 100%. It means high temperature can enhance the rate of reaction. Over a period of time, enzyme will be deactivated. And after some time, when the temperature is very high, then enzyme will be deactivated even at moderate temperatures. Why they are moderate? Because they are denatured at high temperature. They are become shapeless. Here you can see the temperature on one side, x-axis and y-axis rate of reaction. So from zero, as temperature is increasing, the rate of reaction is also increasing because the kinetic energy of the molecule is increasing as the temperature is increasing. So they move more fastly. So when they strike fastly with the enzymes active site, so what will happen with them when they strike with them? They are unable to attach themselves on the active site of the enzyme because they are very fast. So when they strike again, they damage the active site or simply reshape the active site, which is called as denaturation of enzymes. So at high temperature, after this temperature, the enzymes will start denaturation and they will reaction will moving downwards and then it will stop after some time because 40 degrees centigrade is the optimum temperature for enzyme activity or enzyme action. So after this temperature, the denaturation of enzymes will be started. Due to high kinetic energy of those molecules, they will move very fastly, move towards the next slide, which shows the effect of pH on enzyme activity as we had discussed about temperature. Enzymes are affected by changes in pH. The most favorable pH value, the point where the enzyme is most active, is known as the optimum pH. If that where the pH value is best for enzyme action, that is called as optimum pH. Extremely high or low. High means alkaline, low means acidic. Values generally result in complete loss of activity for most enzymes. You can see 7, 14, 1. Neutral, al- alkalinity is increasing, and in this opposite direction, there is acidity is increasing. So this is best pH for some enzymes, and some enzymes have different pH of their pH. It can be 2 in some enzymes, it can be 6, it can be 7. So in this way, they work at different pHs. Here I have shown you reducing or increasing the pH away from the optimum pH reduces the rate of reaction because the concentration of H positive in solution affects the tertiary structure of the enzyme molecule. So it will affect the shape of the enzyme, which will become denatured. And again, it is called as denaturation of enzymes. You know about amylase, it works best between 6.5 to 7 pH. And our stomach has gastric glands and gastric juice is secreted, and gastric juice contains pepsin, and pepsin work best between 2 to 2.5 or 2 to 3 or is so in this way if we want that amylase should work in acidic conditions, it will not happen. Amylase will denature. And same is the case with the pepsin. If we want that pepsin should work at 7 or 8, so it will not work. It will become denatured. So in this way, I would like to say that the enzymes are denatured. So students, it was all about the chapter 5 enzymes. Thank you so much for watching my video. Please like, subscribe and share my channel. Inshallah, we will meet in the next video. Thank you so much.